Greetings everyone, and welcome to the screencast where we provide an overview of our project to develop and deploy a cancer transcriptome analysis toolkit, leveraging our Trinity software, with the goal of assisting researchers in applying RNA-Seq to genetic and functional analyses of cancer. This project is funded by the National Cancer Institute's Informatics Technology for Cancer Research and led by Aviv Regev and myself, Brian Haas, both of the Broad Institute and the Klarman Cell Observatory, and in collaboration with Bill Barnett at the Indiana University Supercomputing Center. RNA-Seq is a powerful technology for studying transcriptomes. In the case of cancer, it can provide a window into the genetic and epigenetic state of a tumor based on the regions of the genome that are actively being expressed. The applications for RNA-Seq in the study of cancer are fairly extensive. RNA-Seq yields base-level resolution of the transcriptome, so it's possible to identify individual mutations. Alternative splicing patterns are readily apparent, making it possible to identify cancer-specific isoforms or evidence of aberrant splicing, perhaps leading to oncogenic transcripts. RNA-Seq can hint at structural variations due to chromosomal rearrangements or translocations that often generate fusion transcripts. Fusion transcripts can sometimes be potent drivers of tumorigenesis, and their proper identification can have significant prognostic value. Another aspect of RNA-Seq is that it can be used to identify other foreign genetic elements that may be associated with that tumor sample, such as from the microbiota or from viruses being expressed. One of the commonly performed tasks in RNA-Seq studies is to reconstruct the transcripts from which the short reads were derived. There are a couple of approaches to reconstructing transcripts from RNA-Seq data. In one approach, the reads are individually aligned to a reference genome sequence using a short read spliced alignment tool that can map reads across introns. Then, the short read alignments, instead of the reads themselves, are assembled into gene structures. A very popular software package for genome-based transcript reconstruction is the Tuxedo suite. This approach is highly effective, but also inherently limited to the quality of the reference genome to which the reads are being aligned. In another approach, the reads can be directly de novo assembled in absence of any reference genome sequence. Reconstructed transcripts can then be aligned to a given genome to infer gene structures for those genes that are well represented by the reference. But in this case, we'll also retain those transcripts that are not well represented by a reference genome, such as due to the genome restructuring that often occurs in cancer genomes, or due to having a foreign origin, such as from viruses. For de novo transcript assembly, we developed a tool called Trinity, and Trinity is a primary focus of what we're planning to leverage and make more accessible to researchers as part of a cancer transcriptome analysis toolkit. As an example of how this is useful, we took a previously published RNA-Seq dataset corresponding to breast cancer cell lines, assembled the reads using Trinity, and examined the reconstructed transcriptomes. Shown here is a heat map that indicates the number of reads found to overlap a fusion transcript junction for validated fusion transcripts in each of the 4 cell lines. Those breast cancer fusion transcripts having sufficient read support were found to be correctly reconstructed as fusion transcripts by Trinity, as highlighted in purple. Trinity reconstructed many of them as full-length fusion transcripts, including several alternative fusion isoforms. In addition, we identified transcripts corresponding to a bovine polyomavirus, which had likely infected one of the cell lines while growing in contaminated media. In addition to applying RNA-Seq analysis to cancer samples, technologies are now available that allow for the study of transcriptomes at the level of individual cells. This is especially relevant to studies of cancer. To give you a concrete example, work done in collaboration with our group at the Broad and published last June had demonstrated the use of RNA-Seq data to study tumor heterogeneity at the single cell level in primary glioblastoma, which is a very deadly form of brain cancer. Using RNA-Seq data, we are able to characterize variations within tumor cells that are implicated to be highly relevant to disease progression and prognosis, such as oncogenic splicing variants in EGFR, deleterious mutations in TP53, and gains and losses of complete chromosomes, known to be the most common genomic aberrations encountered in glioblastoma. Ultimately, we aim to make these types of analyses readily available and easily accessible to any cancer researcher, and it should all just start with a set of FASTQ files from RNA-Seq data. One way in which we aim to make RNA-Seq analysis tools, including Trinity, accessible to all cancer researchers is by a Galaxy web interface, where users would upload RNA-Seq reads, run tools, navigate results, and generate reports using our Cancer Transcriptome Analysis Toolkit. Our Galaxy portal has just gone live as of early January, and so Trinity assembly services are now currently available to all researchers having RNA-seq data and an internet connection. Simply visit the link as shown. We'll be building out many additional capabilities in our analysis toolkit over the course of this project, including mutation detection, fusion transcript identification, expression and splicing analysis, and wide-ranging support for single-cell transcriptome analysis. We have much more to come. For additional information about Trinity or our cancer transcriptome analysis toolkit efforts, please check the Trinity website at trinityrnaseq.sf.net, where we have software documentation, analysis protocols with links to publications, software tutorials, and workshop materials, and be sure to subscribe to the Trinity mailing list. Here we acknowledge the primary contributors to this effort, including Principal Investigators Aviv Regev and William Barnett, and we give additional thanks to ITCR for funding these efforts.